everyone, and welcome to the opening night of Back to Bacteria, Episode 1, The Microbial Menace, your favorite fine theory production starring your favorite microbe. Many of you have met me before, scratch that, all of you have, because I'm right here in your gut. But you probably first learned my name in your freshman biology textbook. Yes, that is I, the bacterial icon, Escherichia coli, but for my dearest listeners, call me Rick. Our topic of the day is bacterial genomics, the study of bacterial DNA, specifically how it can be used to combat antimicrobial resistance. We'll start things off with a brief introduction by our creator, because for some reason she wants to get emotional, and then we'll launch into the learning. Hi everyone, thank you for making it to the first showing of Bacteria. Originally, I wanted to make an omics puppet show for the end of the year bio final as a joke. After all, why would I risk such a large part of my grade on something that seems so silly? But then I realized it was a chance to combine some of my greatest interests: genomics, arts and crafts, and making strange voices. I began gaining an interest in genomics when I decided to try out for AmeriCorps' computational biology camp the summer before 9th grade. I didn't realize I had discovered one of my favorite fields of study. I've continued to explore omics through my school's Guidance Club and the AmeriCorps volunteer program, where I learned that by analyzing gene expression levels in disease versus control groups, scientists could pinpoint what proteins were being differentially expressed, leading them to find potential biological processes associated with the condition. This year in AP Bio, we learn about microbial resistance and its disastrous toll on human lives, as well as the heavy financial burdens. I wanted to see if omics could be applied not only to humans, but also to bacterial DNA, to see what cellular pathways are involved in antibiotic resistance, and what potential solutions there are to inhibit these pathways to increase the capabilities of antibiotics. Thank you, and I hope you enjoy the program. Okay, enough of that. On to our first segment. Our first guest specialist is here to talk about antimicrobial resistance. Give a warm welcome to Pinky the antibiotic. Hi everyone, my name is Pinky, and for as long as I can remember, I've been helping humans fight against nasty bacterial infections. In the beginning, I found the life of an antibiotic fairly straightforward. After all, killing bacteria seems so easy, but lately me and countless other antibiotic pills have been struggling as bacteria mutate to resist our attacks. This phenomenon is called antibiotic resistance. It's a serious global health threat, leading to 1.7 trillion in economic losses annually, and it's expected to cause approximately 1.9 million deaths per year by 2050. One thing that makes the problem even worse is when humans misuse antibiotic treatment. It leads to a higher rate of mutations. When a bacterium develops antibiotic resistance, it spreads rapidly across the environment. This is because resistant bacteria can divide so quickly to create more resistant bacteria. Additionally, bacteria can release plasmids with resistant genes so that previously non-resistant bacteria become resistant. Eventually, there can be millions of bacteria that antibiotics can do nothing against. However, pathogen genomics can help us understand how antimicrobial resistance starts and spreads by tracking resistance across humans, animals, and the environment. Scientists can use genomics to discover new resistant mechanisms, and if we find pathways, we can also figure out how to block them. There are many types of genomic measures, but here are some major ones. There's next-generation sequencing, which is the fastest and most affordable genome sequencing, invented in the early 2000s. Additionally, there's whole genome sequencing, a subset of next-gen sequencing where, as the name suggests, you sequence the animal's entire genome. Additionally, many types of genomics focus on different pathogens, such as bacterial genomics, which focuses on bacterial genomes, and cancer genomics, which focuses on studying cancer cells. Specifically towards antimicrobial resistance, or AMR, these techniques are used to identify resistant genes and develop better ways to control infections. That sounds like a world of possibilities. Now, welcome our next guest, who's certainly a genius, and will be taking us deeper into the world of bacterial genomics. Thank you. Welcome, Helix. From what I understand, you're a piece of sequenced DNA. Yes, sir. Why don't you give us a bit of history on bacterial DNA sequencing? Well, the first bacterial genome to be sequenced was Haemophilus influenza in 1996. It paved the way for scientists to understand bacterial physiology, metabolism, and virulence. It's always influenza. Then, in 1997, E. coli was sequenced, and in the early 2000s, next-generation sequencing was developed, and many more bacteria were able to be used to investigate AMR. For example, methicillin-resistant Staphylococcus aureus genomes were sequenced in 2001 to better understand the mechanisms of drug resistance. Early whole genome sequencing studies also uncover the role of mobile genetic elements, such as plasmids and transposons, which are pieces of DNA that spread resistance through bacterial populations. Statistical genomic methods using large datasets of bacterial DNA sequences were also used to identify resistance-associated mutations, such as the WOC-R mutation, which is associated with vancomycin resistance in S. aureus bacteria. But what exactly are the goals of bacterial genomics? How can this stuff help combat bacterial illness? Well, the most direct goal is to find resistance genes or mutations in bacteria. But scientists can also use these genes, as well as their expression levels, to predict how resistant a strain of bacteria will be towards an antibiotic. Scientists are also learning more about how resistance evolves over time, as well as how it spreads so quickly, even between different people and environments. Well, what tools do they use to do this? Well, AI and machine learning have been commonly used to analyze large-scale datasets, and CRISPR gene editing can be used to validate conclusions by testing to see if genes hypothesized to cause resistance actually do in a lab setting. Moreover, genetics can be used to show how resistant bacteria move between different groups, whether it be from country to country or even animals to humans. Moreover, phylogenetic trees can be developed using similarities between DNA sequences to track how bacteria evolve from each other and how resistance evolves over time. Well, what are some future applications? Some of the most exciting applications of genomics in a clinical setting is precision medicine, an approach to healthcare that takes differences in other people's genes into account to increase the effectiveness of medication. Scientists are working on developing genomic-based diagnostic tests that can accurately detect resistance mechanisms in clinical samples using the sequencing of bacteria infecting the samples or sequences from clinical samples, and can be used to predict resistance determinants. These rapid diagnostic techniques can potentially guide physicians in making informed treatment decisions and optimizing antibiotic therapy to reduce the risk of treatment failure. Okay, now that our introduction and background sections are over, we enter to the real reason we're here. Well, isn't that because of a bio final? Well, partly, but it's also to talk about a groundbreaking scientific study that was published in Nature just this week. In the last year, there have been many breakthroughs using genomic data in clinical settings, and Pinky, Helix, and I are about to share one more. For the first time, scientists have used bacterial genomics to investigate treatment failure in Staphylococcus aureus infections. Inspired by strategies used in cancer genomics, scientists applied combinations of bacterial genomics and antibiotic susceptibility testing to identify and track adaptive mutations that cause antibiotics to fail. But why is this work so significant anyways? Well, this study is especially noteworthy considering how novel bacterial genomics is compared to human and cancer DNA sequencing studies. In cancer, detecting specific mutations can allow doctors to make informed and individual decisions with better outcomes. Similarly, the detection of adaptive mutations, especially in response to antibiotics, can help us understand why antibiotic failure occurs and can help us make predictions about AMR. Really? How? Well, take the example of Staphylococcus aureus, which often gains resistance to B-lactam antibiotics such as oxalicillin due to the presence of modified penicillin-binding proteins. However, oxalicillin resistance even occurs in the absence of this protein. By assessing bacterial genomic data, scientists saw the acquisition of multiple adaptive mutations, such as the development of the TAG-O gene, that drove penicillin resistance. This has potential clinical implications, since genes such as TAG-O can act as biomarkers to help identify treatment failure early, and it can inform potential treatment alternatives. And it's just not Staphylococcus. Similar studies can be done with tuberculosis, pneumonia, and many others. Even E. coli, because I have a few pesky neighbors that I just can't seem to get rid of. Well, sure, I guess. On that happy note, we have reached the end of our segment. Give a special thanks to our guest speakers, Pinky the Antibiotic and Helix the DNA segment. And that's it for today, folks. Here, on episode 1, The Microbial Menace, we've learned about bacterial genomics, the study of bacterial DNA sequences, specifically how it can be applied to solve problems caused by antimicrobial resistance. We took a deep dive into a specific research study involving Staphylococcus aureus bacteria and learned about the applications of bacterial genomics in personalized precision medicine. From all of us here at Back to Bacteria, we thank you for listening and hope you either learned something or were at least mildly amused. We'll see you next time for episode 2, Attack of the Coli.